Hi everyone, welcome back to Pill Talk. We will discuss today about changes during muscle contraction. To begin, let's discuss the foundation of muscle contraction. So, muscles contract when they are stimulated, and this contraction is essentially a physical or mechanical event. However, it is important to note that muscle contraction involves much more than just mechanical movement. Several interconnected changes occur within the muscle during this process, and these changes include electrical changes, physical changes, histological, or we can say molecular changes, number four, chemical changes, and finally, thermal changes. Now let's discuss the electrical changes that occur during muscular contraction. These changes can be categorized into two states. Number one is the resting condition and number two is the active condition. Let's discuss the resting state first. So in the resting state, the muscle exhibit a phenomenon known as the resting membrane potential. This refers to the electrical potential difference across the muscle cell membrane, or we can say sarcolemma, so when the muscle is at rest. Essentially, there is a difference in charge between the inside and the outside of the muscle fiber. Inside the muscle, there is more negativity compared to the outside, which is more positive. This is due to the distribution of ions like sodium, potassium, and chloride across the cell membrane. If we connect two electrodes to a cathode ray oscilloscope, one can place on the muscle fiber surface and the other inserted into its interior. Then we can observe this potential difference. So this state where the muscle fiber is at rest is known as the polarized state, and for human skeletal muscle, the resting membrane potential is typically around minus 90 millivolts. So when the muscle transitions into an active state, the resting membrane potential changes as it responds to stimulation. So this is known as an action potential, which initiates the process of muscle contraction. Electrical signals during this state are also measurable using devices like cathode ray oscilloscopes or computerized polygraphs. So the development and maintenance of the resting membrane potential in a muscle fiber or neuron are crucial for its function. This potential arises due to the movement of ions across the cell membrane. It creates an ionic imbalance. So this imbalance results in a greater positive charge outside the cell and greater negative charge inside the cell. So the ionic imbalance is responsible for the resting membrane potential is maintained by two key mechanisms. Number one is sodium-potassium pump and number two is selective permeability of cell membrane. Let's discuss sodium-potassium pump first. So the sodium-potassium pump actively transports ions in opposite directions across the cell membrane. Specifically, it moves 3 sodium ions outside of the cell and brings 2 potassium ions into the cell. So this process requires energy from ATP. By moving more positive ions outside than inside, the pump creates a net deficit of positive charge within the cell. So this contributes to the negativity inside the cell and the positivity outside, which is essential for the resting membrane potential. So the sodium-potassium pump plays a vital role in maintaining the electrical balance. Number two is selective permeability of cell membrane. So the cell membrane's permeability depends on specialized transport channels that allow selective movement of specific ion. So these channels are often gated, means they open or closed based on specific conditions. So there are two main types of channels that are basically involved. Number one is channels for major anions, negatively charged substances such as proteins. So these channels typically restrict the movement of large negatively charged molecules like proteins, organic phosphates, and sulfates. So as a result, these substances remain inside the cell. It contributes to the overall negative charge within the cell. Number two is leak channels. So these allow the slow passive movement of ions like potassium out of the cell. It further influences the ionic balance. Now let's focus on leak channels, which play a crucial role in maintaining the resting membrane potential. So these channels are passive. They allow the movement of positive ion, particularly sodium and potassium across the cell membrane. So firstly, sodium and chloride ions are found in higher concentration outside the cell, whereas potassium ions are more concentrated inside the cell. So the unequal distribution of these ions creates the condition for the resting membrane potential. Chloride channels remain mostly closed during resting condition, which means chloride ions are retained outside the cell. Thus, the movement of ions through leak channels is primarily limited to sodium and potassium. So sodium ions are actively transported outside the cell and potassium ions are actively transported into the cell by the sodium-potassium pump. However, due to concentration gradient, some sodium diffuses back into the cell through sodium leak channels, while potassium diffuses out through potassium leak channels. So in resting condition, most of the potassium leak channels are open. It allows potassium to diffuse outside the cell freely. In contrast, only a small portion of the sodium leak channels remain open, so less sodium enters the cell. So the efflux of potassium is greater than the influx of sodium, which helps in maintaining and stabilizing the resting membrane potential. After the resting membrane potential is established, with the inside of the cell being negative and the outside is positive, the movement of potassium through leak channels stops. So please focus on this thing. So the inside is negative and outside is positive. The positive charge outside the cell repels additional potassium from leaving, and the negative charge inside the cell attracts potassium ions, preventing further leakage. Discuss the critical role of intracellular potassium ions in maintaining the resting membrane potential and ensuring proper cell function. So the concentration of potassium inside the cell is approximately 140 milliequivalent per liter, which is significantly higher compared to the concentration of sodium outside the cell. So this high intracellular potassium concentration is essential for maintaining the negativity inside the cell, which is a key characteristic of the resting membrane potential. So in a typical muscle fiber, the resting membrane potential is around minus 90 millivolt, while in nerve fiber, it is approximately minus 70 millivolt. So this negativity is largely due to the presence of negatively charged proteins, organic phosphates, and sulfates within the cell. So these large anions cannot easily move out of the cell, and thus it contributes to the internal negative charge. So potassium ions help regulate this resting negativity by counterbalancing the internal negative charge. So if the concentration of potassium inside the cell decreases or potassium is not adequately maintained, the resting membrane potential becomes more negative. It will exceed towards like minus 120 millivolts. So this state is called hyperpolarization. Hyperpolarization has significant implications for cellular functions. It makes it more difficult for the cell to reach the threshold needed to generate an action potential. As a result, the development of action potentials may be delayed, or in severe cases, it may not occur at all. Let's take a closer look at figure, which shows the development and maintenance of the resting membrane potential through the activity of sodium-potassium pump and the diffusion of ions. So the sodium-potassium pump is a key active transport mechanism shown here. So this pump works by actively moving 3 sodium ions outside of the cell and simultaneously transporting 2 potassium ions into the cell. So alongside the pump's activity, the diffusion of ions across the membrane also plays a crucial role. So due to the concentration gradient, potassium ions tend to diffuse out of the cell through potassium leak channels, which are predominantly open. Although sodium also has a tendency to diffuse back into the cell, so mostly sodium leak channels remain closed, so it limits this movement. Let's now discuss the concept of action potential, which is a fundamental electrical process in excitable tissues like muscle and nerve cell. So what is an action potential? An action potential refers to a series of electrical changes that occur in the membrane potential when the muscle or nerve is stimulated. It is essential for communication between cells and the initiation of muscle contraction. So the action potential occurs in 2 key phases. Number 1 is depolarization. So depolarization is the initial phase of the action potential. During this phase, the inside of the muscle cell becomes positive and the outside becomes negative. So this occurs because the resting polarized state is abolished and sodium ions rushed into the cell. So this reversal of charge sets the stage for cellular activity such as muscle contraction or nerve impulse transmission. Repolarization, on the other hand, is the recovery phase where the membrane potential returns to its resting state. During this phase, the inside of the cell becomes negative again and the outside becomes positive. It reestablishes the polarized state. So this is achieved by the movement of potassium out of the cell. And action potential curve represents the graphical registration or representation of electrical activity in excitable tissue following by the stimulation. So it is divided into 3 major parts. Number 1 is the latent period, number 2 depolarization phase, and number 3 repolarization phase. Question arises, what is the latent period? So the latent period is the short interval immediately after a stimulus is applied, during which there is no observable change in the electrical potential of the muscle. So this period typically lasts only 0.5 to 1 millisecond, and it is important to note that while there are no visible changes, internal processes like ion channel opening and preparation for depolarization are already underway. So before the latent period begins, you may notice a small irregular deflection in the baseline on the action potential curve. So this is referred to as the stimulus artifact. It occurs due to a disturbance caused by the leakage of current from the stimulating electrode to the recording electrode. So the stimulus artifact is brief and precedes the actual latent period. So looking at the figure, you will see the stimulus artifact is marked as slight deviation in the baseline. So this is followed by the latent period where the curve remains flat, indicating no immediate change in potential. Let's now move to the second phase of the action potential, which is depolarization, which follows the latent period and marks the beginning of significant electrical change. So depolarization is the phase where the muscle cell membrane potential becomes more positive, moving it from the resting state towards zero and beyond. So the process starts slowly with the membrane potential rising by approximately 15 millivolt to about minus 75 millivolt. So at this point, the rate of depolarization suddenly accelerates. So this critical point is called the firing level, as it initiates a rapid shift in the potential, as you can see in the figure. So after reaching the firing level, the membrane potential quickly rises to zero potential, also known as the isoelectric potential. From here, the curve doesn't stop, so it overshoots beyond the zero potential. It reaches up to plus 55 millivolts. So this phase is referred to as the overshoot. You can see in the figure the gradual rise during the initial depolarization phase, the rapid climb to the firing level marked on the curve, and the overshoot where the potential moves above the zero line. It reaches its peak. Let's now discuss the third and final phase of the action potential, which is repolarization. So what is repolarization? So once depolarization is complete and the membrane potential reaches plus 55 millivolt, the process of repolarization starts. So initially this phase occurs rapidly but slow down as it progresses, ensures that cell returns to its resting state. So key components of repolarization are firstly spike potential, the rapid rise during depolarization and the initial rapid fall during repolarization are together called as the spike potential. So this phase is brief. It lasts only about 0.4 millisecond and is clearly marked on the action potential curve. So second component is the after depolarization, or we can say negative after potential. So following the spike potential, repolarization slows down. So this phase is known as the negative after potential and it lasts for 2 to 4 milliseconds. So during this time, the membrane potential gradually approaches the resting value of minus 90 millivolt. So the third component is after hyperpolarization or positive after potential. Sometimes the membrane potential becomes even more negative than the resting levels after repolarization. So this is called hyperpolarization or the positive after potential. So this phase lasts for more than 50 milliseconds and represents a temporary overshoot before the cell stabilizes. Finally, the normal resting membrane potential is restored. It completes the cycle. To understand the ionic basis of the action potential, let's examine the role of voltage-gated sodium and potassium channel in driving this process. If we want to understand it in a more simpler way, we can divide the graph or the curve into some sections as A, B, C, and D, as you can see. So the point A represents the opening of few sodium channels and the initiation of the depolarization phase. And the point B represents the activation of many sodium channels and the leading to rapid depolarization and overshoot. And the point C represents the inactivation of sodium channels and the opening of potassium channels. And the point D represents the closure of potassium channels and ending hyperpolarization and returning the membrane potential to its resting state. Now let's focus on the chemical changes that occur during muscular contraction, particularly the liberation of energy, which is essential for this process. So firstly, let's discuss liberation of energy. The energy required for muscular contraction is primarily derived from the breakdown and resynthesis of ATP. ATP, or we can say adenosine triphosphate, serves as the energy currency of the cell and provides the necessary power for muscle activity. Let's discuss the breakdown of ATP. So during muscular contraction, ATP is broken down into ADP and inorganic phosphate ion. So this reaction releases energy as shown in the equation. So after that, functions of energy released, we can discuss that. So the energy released from ATP breakdown facilitates several critical steps in the contraction process, which includes number 1 is the separate of the action potential, number 2 is the release of calcium ions from the sarcoplasmic reticulum, number 3 is the movement of myosin heads, and number 4 is the sliding mechanism. Now let's discuss how ATP is resynthesized. So the resynthesis of ATP can be done from creatine phosphate. So creatine phosphate, which is a high energy source present in resting muscles, it serves as an immediate source of high energy phosphate radicals. So in the presence of enzyme creatine phosphotransferase, creatine phosphate donates a phosphate group to ADP. It forms ATP and creatine. So the reaction, as you can see, ADP plus creatine phosphate leads to form ATP plus creatine. So this process, known as Lohmann's reactions, provides energy quickly, but it is short-lived and support muscular contraction for only a few seconds. So once creatine phosphate is depleted, the muscle relies on other energy sources such as carbohydrate metabolism for longer lasting ATP production. Let's discuss the resynthesis of ATP by carbohydrate metabolism. So glycogen is used as a stored energy source. So in a resting muscle, glycogen is stored in the sarcoplasm and it undergoes catabolism to produce ATP. This process begins with glycolysis, which is also known as the glycolytic pathway or Embden-Meyerhof pathway. And during glycolysis, each glycogen molecule is broken down into two pyruvic acid molecules which produce two ATP molecules. So this pathway consists of at least 10 enzymatic steps, each catalyzed by one or two specific enzymes. Hydrogen atoms are released, which contributes to further ATP production later on. So if oxygen is present, pyruvic acid enters the aerobic respiration pathway, which will lead towards the production of significantly more ATP. In the absence of oxygen, pyruvic acid undergoes fermentation to form lactic acid, which produces less ATP but allows the muscle to continue functioning temporarily. Now let's discuss the changes in pH that occur during the different stages of muscular contraction. So in resting condition, the muscle reaction is alkaline with a pH of approximately 7.3. This reflects the absence of active biochemical processes like ATP breakdown or lactic acid formation. It maintains a stable, slightly alkaline state. And during onset of contraction, at the beginning of muscular contraction, the muscle becomes acidic. So this acidity is caused by the dephosphorylation of ATP, where ATP is broken down into ADP and inorganic phosphatine to release energy for contraction. And during the later part of contraction, as contraction progresses, the muscle reaction shifts back to alkaline. So this is due to the resynthesis of ATP from creatine phosphate, which reduces acidity and stabilizes the pH during sustained activity. At the end of contraction, at the conclusion of contraction, the muscles become acidic again. So this final acidity is due to the accumulation of pyruvic acid or lactic acid, especially during anaerobic activity when oxygen supply is insufficient for aerobic metabolism. Now let's discuss thermal changes during muscular contraction. So during muscular contraction, heat is produced. Not all heat is liberated at a time. It is released in different stages. Number 1 stage is resting heat, number 2 initial heat, and number 3 recovery heat. So what is resting heat? Even when the muscle is at rest, it produces some amount of heat. So this is called resting heat. It is the result of basal metabolic processes within the muscle which sustain its basic function. Number 2 is initial heat. So what is initial heat? During muscular activity, heat is produced in multiple stages, collectively known as initial heat. It can be divided into 3 types. Number 1 is heat of activation. So this is the heat generated before the actual shortening of muscle fibers begins. It occurs during the release of calcium ions from the alveoles, which is essential for initiating muscle contraction. It is also referred to as maintenance heat. Number 2 is heat of shortening. This is the heat produced during the shortening of muscle fiber. It arises from structural changes in the muscle such as the movement of cross bridges, the action of myosin heads, and the breakdown of glycogen for energy. Number 3 is heat of relaxation. So this heat is released during the relaxation phase as the muscle returns to its resting state. It is primarily produced during the breakdown of ATP and is released as the muscle lengthens during relaxation. And last one is recovery heat. We will discuss this in the next slide. Finally, let's talk about recovery heat, which is an important phase in the thermal changes during muscle contraction. So what is recovery heat? Recovery heat refers to the heat produced in the muscle after the completion of muscular activities. This heat generation occurs during the recovery phase where the muscle undergoes various chemical processes to restore its normal state. So why is recovery heat produced? After muscular activity ends, the muscle begins to replenish the energy reserves that were depleted during contraction. This involves resynthesis of ATP, reconstruction of chemical substances such as creatine phosphate and glycogen, which were depleted in the previous phases. So these chemical reactions are energy intensive and results in the production of heat. And with this, thank you so much for watching, and don't forget to like, share and subscribe. Stay healthy and stay informed.